Hello, Krishna. Hi. I think you're muted. Just waiting for everyone to show up here. Yeah. There we go. There's Chuck. Am I up? Someone's audio is like there's a static.
Hello, hi sir. Hi.
Alright, how are you doing?
Yes, sir, fine.
Good. Is that you, Mayukh? Your background has a lot of static on the connection.
In my background?
I think so.
Yeah, wait then.
It's a
I'll
little
just
bit
switch off the
better.
fan.
Okay, yeah.
I'll switch off the fan and wait. Is it any better now?
Yeah, it's better.
Yeah, fine.
Okay. Let's see. I don't know if Vinay's going to join us today.
You won't join us?
I don't know. And there might be some other people coming in a minute or two. But welcome to the meeting, everyone. This week we're going to do Google Summer of Code, like here an orientation, and we're going to have 2 presentations. So thanks again to everyone who applied to Google Summer of Code this year. Krishna applied. He didn't get selected, but he's going to join us. And we'll be doing some things this summer. We have other tasks that we have besides the Summer of Code projects. So there's a lot of opportunities to contribute. And as for Mayukh and Ujjwal, welcome to the Summer of Code program. DevoWorm's been doing Summer of Code for a couple years. OpenWorm and more generally, but DevoWorm is specifically since about 2017, and OpenWorm has been doing Summer of Code since about 2013 or 2014. So they've had a lot of students, and so this is part of your community period exercises. So this month until the end of the month is community period. So according to the schedule, you're supposed to be interacting with people, in the organization. And so I encourage you to go to the OpenWorm Slack. I know all of you, I think all 3 of you, the students here, have been on the OpenWorm Slack. And I encourage you to ask people what they're up to, or what if you find something interesting in the different channels, ask and see if there's anything, of interest that you might, talk to someone about. We might, in a couple weeks, we might have another presentation like this to OpenWorm, there's an event called the Office Hours, and that's a little bit different than this. This is where you would go in to the Slack and you would, type in your, if we give a presentation, it would be, you'd type it into the Slack channel instead of presenting it. And the idea there is we want to get people engaged in the OpenWorm community who wouldn't be able to, say, make a meeting like this. So I want to do that too. But definitely explore OpenWorm. OpenWorm has a lot of different components. So there's DevoWorm, which is this group here, and we're interested in development. There's a movement group they're not as active right now, at least not that I know of, but they do a lot of analysis of movement data. So C. elegans moves around, and when they measure, they've been able to measure motion quite exquisitely. They have a lot of data in one of their they have a database of movement data, and so one of their initiatives is to put this all online, make this available, but also to analyze the data using machine learning and other techniques. Which brings me to one of the other groups, and it's a fledgling group. It's on data science, and so there's a data science interest group, but I don't know if I think there's a channel for it. It's not a formal initiative, but last year we had a couple people who were interested in data science and of the Google Summer of Code students, and they worked on various data projects. So we might, there might be some interactions there. This year we don't have a huge sustained interest in it, but we might start to talk about it. So that's what's going on in the OpenWorm community, and there's probably plenty other things going on. We can maybe talk about that next week, like, what kinds of opportunities there might be, or sort of the layout of OpenWorm. I didn't really want to spend a lot of time on that this week, though. So, but I did want to go through a couple of things here as well. Let me share my screen. So you can see my screen.
Yes, we can.
Yes, sir.
Good. So this is the repository where we're going to do the GSOC commit, the Google Summer of Code commits. Every year, I usually have people commit to a single repository, and this keeps everything in order, both for like code reviews, meetings, and like the final submission. So everything would be submitted in this repo. And again, you probably want to push. I think I sent out an email saying that the best policy is to push at least weekly. So before our weekly meeting, you might want to push, or any other time during the week when you find it convenient to push your code. So you can work on things offline, but you really should have like one push a week. And the reason for that is twofold. One is that it allows me to see what you've done for the week. So during the coding period, we'll go through every week and do a shorter, like a review. I'll ask Mayukh, say, what did you do this week? And then he'll tell me. And then we'll go through maybe some issues that he had. And then we'll go through the code. Maybe the pull request is a good way to look at it and see what's been done or what needs to be done. So it's like, I can like sort of audit it as we go along. So this is what we're doing. This is what we need to do. And then you'll have other things going on that you may or may not push right away, but it's all there. The other reason is because you want to have it in one place. So eventually this is all going to be this code base is all going to be submitted as part of your final project. And so your final project submission it's got to work. So they've got to be able to download it and run it for you to pass. And the other thing is that you have to have everything sort of, front facing. It has to be open. So it has to be in one repository, and it has to be, annotated properly and everything. So, and I mean, that's just, presentation wise. So if I go into this repo, there's nothing in it right now, I don't think. But like, say, Ujjwal was done with his project, he would have all of his code in this repository. It would be pushed weekly, and it would be there now. So now he's ready to submit. So he would maybe create like a README. There's a README here. And then it would give instructions on how to run the program. We'll get into how to submit projects later. But the idea would be that you'd have this all in one place. So if someone comes 2 years from now and they want to look at this project, they can simply, go into the README, look at it, run the program, and then ideally they'd like to maybe contribute to this project. So if they want to contribute 2 years from now, they've got to be able to understand what went on 2 years before. And so that's why you want to have everything pushed to one place, have it well documented, and then be able to, have people access it in the future. So that's the reason we want to have it all in one place. And so I suggest you fork this. I think I saw that Mayukh forked this already, probably Ujjwal too. Well, we only have one fork. But to fork this to your home account, and then, push your updates once a week. And it doesn't have to be everything that's new. It's just the stuff that you want to kind of put up, that you think is ready to go. And then, you can update it later with subsequent pushes to the main repository if you need to, like, fix anything or add things. And that's fine. So that's one part of this. The other part of this is I'm starting this definition and concepts. So we might talk about some concepts, especially later in the projects, that we may need to define. So we might come up. I was working on a couple here that we might talk about in later in the summer. Things that, well, I'm not going to get into what they are now, but basically you're going to run into things that we want to define. Maybe we want to take notes on. Maybe they're like concepts if you look into a new method that we want to like put, kind of annotate it for future work. And so this is where we would put all of that in definitions and concepts. So it'd be like meta features is something we were talking about in the, there's this blog post that I sent Mayukh on pre-trained models when he was applying. And that has this term meta features. And so that's not something that's in the literature. It's something we've been talking about off and on in the group. And so this is sort of a notepad for this. And so it gives people an idea of, maybe things that you maybe want to develop further later down the road, but at least to have a record of that. So they're just in Markdown files. So if you want to create your own stub here, your own definition, you would just create a Markdown file in this directory and then put your notes in Markdown, and then we can refine it later. So that's what you're going to be there. That's where you're going to be working, and we'll probably add other things into this repo as well when we're moving along. I don't know what the best way to organize this. Best way to organize this is to have one repository for each project. So OpenDevo Civil Integration is Ujjwal. Pre-trained models is Mayukh. This is for everyone. And then the README gives you, some idea of what's going on in our GSOC group this summer. So these are the project ideas. Check out DevoZoo, join the Slack channel, and then we'll be adding things as we go along. We have another weekly meeting, Fridays at 4 p.m. UTC. So that's a more informal group. So if people are interested in joining in then, It's a different Hangout link, which is in this README. You might want to join in. It's just a more free-form meeting. If you have questions or concerns, you can talk then. You can present things. If you want to present like a demo, that's a good time to do it. So we have that time as well. But usually we'll meet once a week. We'll meet at this time. And this week I want to devote most of it to GSOC, but we'll spend some time, we'll spend a little bit of time at the beginning of each week reviewing the project and then seeing where we have problems. And then if we need to meet longer, we can meet either probably on Fridays for more involved, if we need to debug something or you want to present a demo, you can present demos in this meeting too. We just have a little bit more formal presentation structure for this meeting. Like if you want to present something related to your project, tell me. I can put you on the schedule, and then we can, there's Steve McGrew. All right. Hello, Steve. We're doing an introduction to Google Summer of Code, so you might find this interesting. I was going through some of the technical details. Let's see. So the second thing I wanted to point to is this DevoWorm, or this DevoZoo interface, and this is the link to this interface. So this is something that's been created over the last couple of years. It's sort of a guide to DevoWorm, and specifically like data science and maybe some educational information. So actually Ujjwal is going to be working on updating this summer, so that's part of his project. But I wanted to show people here. So you notice that up at the top we have these tabs, and they have like DevoZoo, DevoNotes, DevoMethods. And there are a couple of icons. This is OpenWorm. So this goes to the OpenWorm website. This is the main website. This is the OpenWorm movement database. So I told you about this movement database. There's a lot of movement-related data there. There's a lot of videos. So this is something that is ripe for machine learning. You might check it out if you already have a project. It may not be too relevant to what you're doing in the summer, but it may also be very interesting to do benchmarks with. They're adult C. elegans, so let's actually look at it for a minute. They're adult C. elegans worms, and they have them moving on like an agar gel. They usually have them in a plate, and they observe the movement, and they do things like looping around and wiggling around in the gel, and so they're able to actually pick up, track that movement, and they have all this data here. So you can find experiments by movement feature, find experiments by genetic strain, and then you have they're all YouTube videos, so you can pick YouTube videos and segment them. So that's available as open data as well. And then this DevoZoo then has a couple of parts. So first of all, there's this page here, which is a basic biology references. So this may need to be updated a bit, but I always try to keep a set of references especially for Google Summer of Code students who want to learn more about the basic biology of C. elegans. So some of you probably, some people in the meeting know quite a bit about the biology of C. elegans, but other people, the Google Summer of Code students perhaps, know very little about the biology of C. elegans. And so this is an attempt to get people up to speed about this basic biology. So it's not intended to be exhaustive, but it gives like some links to different resources here. And I would recommend the students read through some of this if they have questions about the biology, they can go to this place, and we'll be updating it over the summer probably. But it's a good place just to get a good start on this. The other thing I wanted to show, let's see. Oh, this is not what I wanted. This DevoWorm mute. Oh, DevoWorm. No, that's not it. So I think this is the DevoZoo. Okay, so this is the DevoZoo. This is the link to the data. So what I did, I've done this about 2 years ago, and it's been ongoing. I've been adding to it. Is this DevoZoo, which is it's a collection of data from different species, different biological model systems, but also some other artificial life systems. But the thing I wanted to bring your attention to are these data sets for different types of embryos. So the focus is C. elegans, and these are the data that we have that we've done for different these links here at the top. Cell birth and death timing differentiation tree, cell position. These are analyzed data sets. So these are data sets that come in sort of like a CSV file or an Excel, kind of a spreadsheet table type format. And they have their numeric values, and they're things that you can use to either label raw data or just have as data about the developmental process. These other links here down below are actually movies that we have that are available for people to download and take them apart into frames and then do some sort of analysis on them. Again, I need to update some of these sources, but the sources are all local to like a Google Drive, in OpenWorm. But you'll want to look into these data, and they're available for you to play around with. There are other embryos as well. So you have some microscopy movies for ascidians, which are a marine invertebrate, and that's an interesting data set to look at. There's Drosophila. We have an imaginal disc. I think this is the a high resolution drawing, and then this is fused cell embryogenesis. So there are a lot of sources of data here, and these are even spider embryos. So there are a lot of different sources of data for embryos and things like that we have. I don't have a lot of annotation on them right now. Maybe this summer we'll be working on some of that as part of Ujjwal's project, updating the sort of the annotation of what each thing is. But also it's a good opportunity, I think, to get, Mayukh was asking me what kind of data we have. These are really some of the data sources that we have, and we can add to them as we go along. There's always stuff coming out that's new. So that, and this is a resource, I think, for the community, more than just for like Google Summer Code students, but Google Summer Code students will find it useful. Finally, I want to get into this. I want to talk about this repository, and I'll give a link in Slack later. But this is something for the community period. So you're in an open source project. This is the whole point of Google Summer Code, is to train people in open source. And so I have a repository here of different readings. And this is for people who are not in Summer Code as well. This is a repository for all sorts of different readings on open source and open science. So there are a lot of things in here. But I wanted to highlight these three at the bottom. These are books that are useful for producing like open source software, practicing open science, and then data science. So those are probably three very good references for the students in terms of like learning something about how to produce open source software, how to market it, maybe even, or how to make it public and make it citable and things like that. So I would recommend that if you're a Google Summer of Code student, or even if you're not, that you take a look at those references. And it's a good educational repository. We also have other readings on open data and social media and licensing. So these are other things that you might have questions about as you go through the summer that will, you may be able to answer them through reading that, or we'll revisit those resources as we go along. But it's definitely something that they're not going to tell you. There's no course in this, as far as I know. You have to kind of learn it from books and from like people's personal experience. So having some having some facilitating some background on that is good. So that's all I wanted to show as kind of an orientation to Google Summer of Code. Did we have any questions about anything? Okay. Any questions? No, we didn't have any questions. We'll revisit these things, of course, like I said, in various channels. And good luck with everyone's project. And Krishna will, again, like if you attend these meetings and you see something you find interesting, please let me know or let someone know that you're interested in it, because we can, eventually we'll find something that's maybe something you can take and make your own. So I just wanted, before we get into the presentations, did we have any other comments or announcements from anyone else? Dick or Steve, did you have anything you wanted to mention? Yeah, I forgot to ask at the beginning of the meeting, but if we don't have any announcements or comments, or why don't we start with the presentations? So we have two presentations. One is Ujjwal and one is Mayukh. So who wants to start first?
I can start first if you want.
Okay, go ahead.
I'll start first then.
Okay.
Yeah, I'll present then.
All right.
Hold on. Hang on a second. It's just. Yeah, is it visible?
Yes.
Okay, can you see it right now?
Oh yeah. Got it.
Yeah. So hello everyone, I'm Mayukh, and just a brief intro that I'm a 19-year-old guy, and I've been doing deep learning since like the last 2 years. And so my project is like basically I'll have to train deep learning models on like basically a lot of data which is there in the DevoWorm and the DevoWorm repos. So yeah, that's it. So I'll just start with the timeline. I obviously start with the community bonding period, and like I just try to get myself familiar with the underlying biology, because like I'm not really sure about how the worm works or anything. So before I get to like classification or anything, I'll have to learn how the cells work, like to get the right idea of the images or the data, whatever it is, in whatever format I'll get it. And then I'll just clear out all my uncertainties regarding the project. I'll go around with the community probably and ask like what kind of a model do they want, or what kind of prediction would be the most helpful to them, because like it should be helpful, right? Yeah, then. So coding period begins, and then initially I just look for hurdles like unbalanced classes. Like I might just have to like classify between classes like class A and class B. So like if one of them has 200, one of them has 500, so the model would be biased. So I'll have to find ways to balance those images. So I'll have to like look for those possible hurdles, and then I'll start testing out image augmentation. Like this is the part where like thanks to like because of image augmentation I'll have to actually know the biology, because like sometimes orientation matters and sometimes it doesn't. So I'll have to do a bit of reading on some of the things that are there and produce I'll start out with my testing with small dummy data sets because it's faster. When I do something on 5,000 or 10,000 images, it'll take me time to train the model and actually see what's going on. But if it's on 200 or 300 images, so I can prototype faster initially and check whether or not it works well. Yeah, so that would be week 1. Yeah, so week 2 would be I'll start after by week 1 I'll get a good idea of the data that I have and what do I have to do for the community with the models. What does the community want? So I'll just start selecting out possible architectures for the models, and then I'll just put down the pros and cons of each architecture for each specific data set, and we can discuss whether we want speed or we want accuracy. Obviously a bigger model would give you more accuracy, but sometimes speed would matter as well, because if it's hosted in the server, it might matter. It depends on the capabilities of the server where it would be hosted. Yeah, so you can read this as far as intuition goes, we can go for a deeper architecture, a bigger and maybe a 60 MB, 70 MB model. If accuracy is important and we have a lot of images from a lot of classes, but if the dataset is relatively small with I don't know, 3 or 4 classes, then it will be a smaller architecture because it's faster. Yeah, then again I'll test those architectures on a smaller dummy dataset. Why dummy again? Because smaller means faster testing initially. Yeah, then the models to be trained first will be chosen based on 2 factors, usefulness to the community and availability of data. It would be the logical end of both of these. It should be first thing should be it should be useful to the community, and then there should be the data available, whether or not in the DevoWorm websites or maybe outside. I can even source it from some other websites other than DevoWorm if required. I don't think it would be required because I've been going through Bradley's links recently and we've got a lot of data to work with. So we'll see to that later on. Yeah. Then weeks 3, 4, and 5. It would be these would be the main weeks. I'll just test out the architectures on the small slice of the data and compare the training metrics. We'll look at the training metrics and we'll compare which one's doing better, which one's gaining higher accuracy, and then I'll try to submit the initial training metrics on the evaluation report, and that's the end of phase 1. So the phase 1 milestones would be finalizing on the architecture, testing out the augmentation techniques, after discussing and after learning about the cell biology, and then I'll just train the models at least once and just look at how they're performing and find a few possible ways if I get extra time to further improve the accuracy. That will be basically phase 2, to improve the accuracy and to get it to that peak. So then phase 2 starts, and until now the models were trained on basic default guess hyperparameters guess learning rates. But now I'll go for different libraries Tool, with which I can basically optimize the parameters learning rate and batch size and all that. We can actually optimize those and get even more accuracy if required. If they have already not peaked, we'll just use those to peak out. Yeah. So then we'll just train the finalized models and everything would be done, most of the part would be done by week 7. That is roughly the first half. And the second half, we'll just move on to benchmarking and helping Ujjwal. So the second part would be I'll just benchmark the models and make sure they're performing well, and I'll probably try and get hold of some more testing data which the model has never quote unquote seen before, so that we can just test out whether or not it works in real life after being deployed. Or initially I'll just test it in a very scratchy way on my own Python files, but later on they should also work in the website. Yeah, I'll update the blog as well. And then phase 2 milestones, most of the work will be done by now, train and save all the models with best possible accuracy on the test sets, and then push some of the notebooks which might be helpful to the community. Bradley we have been talking about pushing some of the notebooks. Some of them are already there in the OpenWorm education repo. I think it's there in that MLDL tutorials folder. I want to push more well documented notebooks which might be helpful for the community, which will come later on. Maybe after 2-3 years down the line, someone wants to start off with what I'm doing, so it will obviously help them, because I'll obviously include Markdown and stuff which would help them. Yeah, then phase 3 is just mostly wrapping up. I'll just start helping Ujjwal in the process of integrating the model into a GUI backend. And I've been actually talking to Ujjwal about this personally, like since we've been selected into GSOC, we've been talking on Telegram based on how he's doing things, and we've been talking, yeah. Then other than that, I'll obviously write documentations like so that like suppose someone doesn't want to use the GUI and wants to use a command line tool to like classify thousands of images at once. So it won't take a lot of time to make a command line tool like that, which would help them. Like maybe we'll just feed it a folder name, and we'll classify all the images within the folder, and it will just return a CSV file with the file name and the classified value, like label it, Yeah, so make a presentation on the pain models and how they can be used by the community. Yeah, then wrapping up on week 12. Make sure that there are no errors. Help as much as possible in integrating the GUI and update my blog with the progress pane and write about how it can be extended further. Yeah, I actually plan to extend it further and like make it a more robust tool with Ujjwal so that it's a real tool which can be used by the community, which is actually useful. Because yeah, that's my thing. Like I try to make things useful. Yeah. Then workflow summary. Like this is how things would like. This basically sums up what I want to do. Like how I want to approach the problem and how I want to like solve it. You guys can see it, right? Yeah. So have a good look at it. Maybe we can have a good look at it after the presentation ends. So yeah. Final question. What kind of a model do you guys think would be of most help to the community? What should we do? Like, what do you guys want? Like, I've been exploring the data since yesterday, so I want to ask that what kind of model is helpful? I've been exploring the data. Some of them are like, I don't know. It's maybe because I don't understand the biology, but that's a different story, right? So I'll have to first learn the biology. And what kind of model do you guys want? Like a classification, like do you have to classify or like look at a cell and tell which stage of a life, like which stage is it in? Forgive me for my, like I don't know the jargon in biology, so I don't know the right words. Yeah, so what kind of model do you guys want? Like what does the community want? I just want to know that. Anyone?
Well, yeah. So I guess maybe anyone can answer what. I don't know if it's easy to answer right now, but
no, like you guys have a better idea than me, like in the community, because I've been very new, so I don't really know.
Yeah.
Like what kind of thing would be useful? Like,
so
do you guys want something that detects a certain event in the cell's timeline?
Well, I guess we can probably.
Hello?
Yeah, we can.
Am I audible?
Yeah, you're here. Can you hear me?
Yeah.
Okay, yeah. So I guess it's kind of a hard question to answer right off the bat, but I think it's definitely something that would capture the process of like cell division in an embryo and some of the some of the processes that are going on during the, like if you watch the movies, you'll see that there's it's not like a typical machine learning data set where you have static images, and in a movie you have like, it's more like a movie where you have like a flow of action going on, and you have things that you might want to. Yeah, it's hard to classify in a discrete way sometimes. But I think that's a question that we're going to have to think about.
So you might want, like, you guys want things which are based on time-based data, right? That's what I understood. Like, it should be time-based mostly.
Yeah, well, the developmental process is
like most of the stuff that is
independent. Yeah.
Yeah. So, like, what do you think? Detecting certain events in process, would that be helpful? Detecting a certain event, like when it goes from one stage to another. Maybe it goes from gas cooler to L1 maybe, or something like that in the C. elegans form.
Am I audible?
Would that be helpful to the community or researchers?
Somewhat.
Yeah.
The movement of the cell.
Yes, Krishna, you are.
You can also predict the movement of
What?
Predict the movement of the cell.
If it can't
Yeah, but for movement you mean like tracking. You mean using tracking.
Am I audible?
Yeah.
Let me try to explain it
Yeah, yeah.
as simple as I can. So basically, you are going to try with some movies which are done microscopy movies, which are will be there, and you have to somehow,
Yeah.
as far as I understand, you have to somehow segment these movies based on and make a unsupervised model. So basically, you have to split the movie which you get into number of frames, and then you have to what hand label it or whatever you
Okay,
think
I get it. I get it.
some amount, and you have to do a segmentation part.
I think one model is not sufficient. You have to make one for segmentation, so my understanding, and one for analyzing and then predicting the movement of
You're
the
not loud enough. Something's wrong with your mic. Like, can you be a bit louder?
Can you hear me? Hello.
No, it's like not loud enough.
Why don't you can type it into the chat as well. That might be better.
Yeah, you can type it in. We can just wait.
Because the mic isn't working.
Yeah, sorry, voice is not.
Yeah, it's so
Yeah, yeah, you can type it down because
Yeah.
I don't know, something's wrong with your mic maybe, I don't know.
So Steve McGrew asked, ultimately, I think we would want the model to be able to generate a movie so we can visually compare with a real movie. So, let's see.
Oh.
That would, yeah, so basically, could you build like a model that would be
Predicting
like
cell movement. Krishna just wrote predicting cell movement. So yeah, I think for predicting or tracking cell movement, we don't really need deep learning. There's this library called OpenCV. And using that, we can actually predict movement, or we can use things like optical flow to predict, not predict, actually track and extrapolate movements. Like if you can track the movement, we can definitely extrapolate it, right?
But basically, you want to do you have to track the cell the nucleus of the cell that you have in the movie that you have provided with, and you have to somehow predict how this gonna move further based on some data. So you have to first find out where is the cell,
Yeah, we can.
the point of the cell,
We have
and
to
then you
basically
have
track and extrapolate. That's the idea. Track and
yeah,
extrapolate.
but the data set that you are going to get is not that easy to that you can go ahead with OpenCV or
Exactly.
something
It's
like
like
that.
the labeling would be a pain, so I'll see to that.
Yeah,
But I think so, Bradley. I think I should ask around the community, right?
I will provide you some labeling tool which will help you. Like there are some open source labeling tool which just you have to draw some borders and it will label it
Yeah.
all for you and form XML files. So this can be very helpful for you.
Yeah, you can drop the links on Slack maybe, or just you can DM me on Slack
Yeah, sure.
instead of dropping it here. So that will be better, right? To keep a place to store it. Okay, wait. Steve wrote something. The model should also start with some assumed principles and predict implementation based on those principles. That way we have a way to test assumptions. I don't get it.
well, it should start with some assumed principles. So, and predict movement. So
Assumed
what he's saying
principles.
is that you might start from what they call first principles. So you might have you might build a model that has assumes certain aspects about cell division. So with C. elegans, we know that the lineage tree, there's a division that occurs. We know when the divisions actually occur. And so we can, just kind of build that into the model. Like, for example, if you track the cells and you sort of, produce a segmentation of each frame and you build reconstruct the movie from that, how closely does it match what we know about the lineage tree? So how closely does it match the timing of known divisions? And so, the thing would be, you'd be able to
Okay, like a way of benchmarking.
Yeah, sort of like that. But
Yeah.
you basically use the information you know ahead of time, and then does it violate that knowledge? So they use first principles models in chemistry and in physics a lot, where they want to assume certain things about molecular laws or physical laws, and then you just basically match it, say does it violate any of these conditions, and so you just
So we want to like predict its embryonic stage or its specific life stage, and other than that, we can also like predict its movements. These are the two prime things that we have to look for, right?
yeah. Yeah. Well, I mean, it's just that we want to have like
So
something that, like, instead of just relying on.
something that does something which can help us, right? Something which can make our lives easier, right?
Yeah. Yeah.
Yes, something. Okay, I get it. I'll have to explore the data a lot more because there's a lot of stuff and I don't understand what's going on in the photos because I don't understand the biology much. I'm really loose on that. I tried studying about the C. elegans worm like 2-3 months back while I was studying the proposal, while I was writing my proposal, so that might help, I guess. I will see to that. But everything is based on those, like that specific tribe folder, right?
Yeah, I think those folders that I sent you a link to will give you a good overview.
Yeah,
Like,
so the thing is that I'll have to, things are going to take a slightly more time than I thought because I'll have to label them as well.
well, you don't.
And to
You
label,
don't necessarily need to label them. Like, well, I mean, ultimately we'd like to, but I think the primary thing is to get like sort of the features like division and motion and things like that down, and then we can worry about labeling them because the labeling. C. elegans, one of the reasons we work with it in this group is because it's the divisions are deterministic. So if we have like just units, we know like cells are dividing and we can identify them, say in space, we can label them. The labeling is less, sort of the labeling is already there. You just have to like match it
no,
up.
I mean, like to train the deep learning model, I need labels, like I need images and I need labels to train it, right? So
Yeah.
I need some resources. No, I can actually label them if required because I have actually done that. Like I think Bradley has seen that repository in which I classified the life stages of the C. elegans. You have seen that, right?
No, I haven't.
It was in the, yeah, it's there, right? So in that, what I actually did was I actually studied a little bit about the embryonic, like the embryogenesis of C. elegans. And so I studied and I understood like this stage, like there are 4 stages, right? So I understood what each stage looks like. Then I just watched a whole time lapse and I cut it into specific timestamps. Manually means like I just watched it and through my own understanding, I cut it into 4 specific timestamps where I thought like, yeah, this is stage 0, this is stage 1, this is stage 2, this is stage 3. So after that I cut them and I split them into frames and did all sorts of stuff. So something like that. That's what I mean by labeling initially because I didn't have any data to work with. There's not a lot of microscopic imagery data that's relevant to DevoWorm online, I guess, as far as I've seen.
Well,
Other than the drive folder that Bradley gave me. Like,
there's a lot. Well, there's a lot of it online. The question is like the quality of it.
Yeah, exactly. Because quality matters a lot. Like 50% of the model's quality would depend on his data.
Yeah.
The rest is the model's accuracy. Yeah, so it's more of a data science problem. And I can handle it for now.
Okay.
Yeah, I'll have to explore the data a bit more.
Okay.
Yeah.
Did you address Krishna's question? You can use adversarial networks for predicting flow. So.
Adversarial network, I've been working on adversarial network lately and I've done like a small project and all. We can use it, but it's going to be a bit more costly on the servers, if you may ask.
Yeah, that might be a problem you'll run into too.
Yeah, they'll be very heavy on the servers. And the thing about adversarial networks is that, see, in microscopic images, as far as I've seen, resolution really matters. Like the images have to be really crisp, right?
Well,
the details.
Oh. Yeah, I mean, depends on. Yeah, it depends on the different types.
Yeah, mostly the details matter. But the thing about adversarial networks is that the networks that we can train on Google Colab and all, like even though the GPUs are really powerful, they return really low resolution images. Like if you want a 128 by 128 image, and if you want to train it on some data set that is above 10,000 images, then your 25 GB of RAM fills up. So we'll have to be careful about that. There are certain computational limits, but we can definitely try it on as a second priority after making the primary models, which would be of most use. As I said in the presentation that like it will be based on the need of the community and the availability of data. If both are there, I'll do it first.
Okay.
Yeah.
Steve McGrew had a comment. One way to think about the embryo development is one cell divides, and then its progeny divide, and so on. Various stages of cells differentiate into different types. They also move to different locations. Their individual destinies depend on the types of cells that surround them and their histories. At the end of the process, there is a well-organized embryo. So a model should probably begin with a set of principles that determine division, differentiation, and movement. So those are the kinds of things I think we mentioned in the blog post, and I think those you might call those meta features, because they're not. I mean, you want to get the cells because you're going to need to know like reference points, but you also want to look at like what's going on between the cells or like within the embryo as a whole. And so those are things that you might not use. You might have like one. You might have one analysis where you track like cell centroids and look at their.
Yeah, I have actually worked on object tracking before.
Yeah, and then another.
So I can definitely try tracking.
Yeah, another like sort of analysis where you look at like different parts of the cell, like alternate to what the cells are doing. What are there things in the images of the embryo? Is there movement that we can classify? Because there's going to be a lot of movement going on. We don't really know.
Ujjwal wrote something. Yeah. Ujjwal says that we need to segment it first and then predict all the features. He's right in a way. Segmentation would definitely help.
Yeah.
Right. Yeah, segmentation would help my models a lot. But I don't really need it.
Like if you are not segmenting it first, so like you have to somehow like if a video has let's say the video is of 4 to 5 minutes microscopic video, so you have a lot of frames in that, so you can't label them every frame, and you can just then segment that this, annotate that this is the cell and there is a cell, and that kind of division.
Right.
So you have to first unsupervised you have to segment the cell frames in unsupervised manner, so that you have all the frames segmented. Then you can apply like you have XML points, so that you can apply where the cell parameter is, and then how the motion is taking place. You can see like how the XML points are changing in every frame. So then you can try finding out that meta parameters that Steve sir has just said, and then after you are in the position to predict something.
Yeah, we can segment it, but for like prediction, segmentations are not always necessary. I've been talking about it in my proposal. That we are seeing that, right? Using parallel models.
That might work in this case, but like you have to first see like data that you are going to get is not that simple. Like it is going to be with different shades. Like it is not going to be always a black background. They are going to see different shades. The parallel modeling might cost you much than the segmentation modeling, and even the results are closing.
I don't get it. Like you mean non-black background? What do you mean by that? You mean noise?
Yeah. It has lots of noise, and it like sometimes the background becomes white or pale or something like that. It's not always black, or at least in the case of passive area.
So we'll have to find a way to segment it then first, like to extract it and to remove the background basically. Yeah, then segmentation would be important. Yeah. So if required, I can work on the segmentation as well, but that's something Ujjwal has been also doing, right? If I'm not wrong.
Well, I know that Ujjwal has done some stuff with it, and then also Vinay has done a lot of work with
I've seen Vinay's work. Like it's like a lot of things, a lot of stuff is there.
So I mean that's.
So I want to ask a question. Like, using the segmentation tool that Vinay actually made, did he actually generate some data? Like, is there a data repository where Vinay kept some data after like segmenting and stuff?
Well, he did create a tool. I don't know if you've seen it. The open.
Yeah, it's some Heroku I've seen.
Yeah. So that's sort of the output of what you get. So we didn't do like analysis of data, but we did have it, where you can upload something, and if it's, there's some, it's a little bit touchy in terms of like the input data, but you can get basically a list of centroid positions, cell sizes and things like that. So it's like a couple of parameters that it reports if you upload an image. The problem, of course, is that we don't have like those meta features. We can't get, it's hard to really kind of extend the model to things that aren't like just, images that aren't just right. So that's kind of where we are with OpenDevoCell, is that, you can do this sort of stuff, but it's a little bit brittle in terms of the.
But I think that would be a little bit of a limitation on my case as well, because like we have to actually input specific images, like specific types of images at least for the models to work.
Yeah. I mean, it's always gonna be tricky with that. Like, you may have to do some pre-processing.
That's where segmentation comes in and pre-processing comes in, or decomposition.
But that's something we're kind of working on independent. Actually, Ujjwal is going to present on this topic or some of the other things. So, thanks, Mayukh, for presenting. I wanted to move on to Ujjwal.
You're welcome.
And sure. We're going to go over 10 o'clock. So if you have to leave at 10, you can leave. I'm going to have this on as a recording. So why don't you present Ujjwal?
Yeah, fine. Yeah.
We're going to...
yes,
yeah. Go ahead and present. We'll continue the discussion about this, like maybe online or in subsequent meetings. So we'll have more to say.
Is my screen visible?
Yeah.
So yes, so I am Ujjwal, and many of us like I have been since in the group for 1 year. And my project title is Open Data Set Integration. So basically, it is a kind of project where I have to do all the machine learning models that we have built so far and improve these models and integrate them in some sort of web app and integrate them with the Python libraries so that we can. So that the main goal of Open Data Set comes to increase the reach of DevoWorm ML, what work we are doing here, and to make it accessible to more wider community. So keeping that in mind, Open Cell comes in. So just a brief introduction. So this project will focus on improving the data science and the machine learning infrastructure of DevoWorm Group. The work will focus on the extension of some of the projects that are completed by Siddharth in 2017 and Vinay in 2019. The two aim is to improve the Open Cell web interface and to improve the segmentation techniques overall. While the Open Cell interface has been implemented as Heroku app, we would like to develop a dashboard for the interpretation as well as tighter integration with DevoZoo, collection of the open source microscopic data. And the third aim was to build Python libraries, and so that these kind of models which we are building this year or which we have built in the previous years can easily be used by the researchers all around the world. So jumping to community bonding period. So yes, I am getting involved in the community and talking with mentors to decide goals. This is one of the meetings where we are going to decide goals for the GSOC project 2020. I have taken reviews from many community members like Mark Worth sir and GY sir to get more and more inputs on what we are doing and how we are going to do it. Deciding color scheme and like how the UX of the open division is going to be look like is one of the major part of community bonding periods. So like I will be posting different color schemes, different design patterns in the DevoWorm ML group and in the group of GSOC this year, so that you can give your suggestions. So like we want it to look best. So more and more sessions we are welcome. Setting up the code base, identifying requirements and setting them up. Like there are going to be a lot of requirements for this project. Like we have right now, I'm thinking of using Django framework, but as the community period progress and might decide like what he want to do, like what kind of tools he's going to use, so we can change that suitably. And so yes, setting up that code base and identifying and creating a whole code base for that. So one of the most important parts are the phase and their goals. So I'm going to discuss that in this slide. So phase one goal is basically working on the front end, completing the front end. part of software and creating website, web pages, dashboards. These are like front end part where these things are going to be static, have to be completed, or designing portion, and should be compatible with the coming models. Connecting to them to a framework like Django or Flask, Node.js, like this will be decided as the community period progresses. One of the major goals for the phase one is like I have to work on C.elegant models and see if I can improve it. I have working since past week on C.elegance model, and I'm thinking like there is something that we can improve in C.elegant model using GANs and something like I don't know how many like there's a new library which comes, so it is also giving a good results in C.elegant model. So I have to work on that and give concrete results like whether I'm able to improve on it or not. If not, if I have improved, then I'll be using that model to integrate in C.elegance as when a model will be integrated. Phase two, successfully integrate all the models of the web portal. By now, we are focusing on three major models that are C.elegance, general image segmentation tools, and statistics tools. So, statistic tools is basically what Steve sir is also talking about. Like, you have to determine like what is the diameter of the cell you are dealing, which you have in the image. What is the diameter of the cell of the in the image that you have uploaded, what are their different meta parameters, so you can see them, you can try to adjust them, zoom in, zoom out, so like it's going to be a kind of photo editor. So yeah, that statistics tool has also been built, and it partially depends on the project 14 as well, because whatever the model is going to train, we can also integrate it, and whatever models we have right now, these are going to be integrated. Phase three is the final phase of this year project. So, making all machine learning models ready to be deployed as DevoWorm Python library. So, this is one of the major goal of this year. Like we are going to make Python libraries for all the project and most of the data sets, so that the researchers around the world can directly use our models and our data set in their Colab or Jupyter notebooks, and finishing the portal with dedicated pages for DevoZoo datasets. So yes, DevoZoo datasets also needs a fresh look and fresh renewal. So I'm going to work on that. I'm going to create a dedicated DevoZoo portal for DevoZoo dataset, which have each individual pages for all the dataset where a person can go and have a look on the website itself, like what this data looks like before downloading it. It will give him some good knowledge about what he's going to download. And also, like we are also thinking that in the portal we are going to give some collab notebooks link. For example, like in the Mayukh, they have just give their suggestion that he's going to upload some notebooks. So it will be great if he is going to upload that notebooks in Jupyter or collab format, so that we can directly link them to the portal. So in a read only format, so that person can know about how the pre-processing is done, how the image is pre-processed that he is going to upload, like all the all the algorithms that we are using to segment his image. It is going to increase his knowledge, and as we are making it more and more open source, people are going to engage in it and start maybe start making pull requests or some issues in the GitHub repositories, so that we can improve it. So like to keep the track of project goals this year, like I have like there is an open source software known as McGrew boards. I find it very fascinating, and I'm using it in one of my other projects. Like I'm also associated with Midas Lab. We work on natural language processing projects. So we heavily use these boards there. So I think that might be good for you. Just let me show you what it is. So yeah, so these are the boards. So these are the sticky boards where you can create your different project layouts, project flow. Like I have created a small project flow for OpenDevoCell, like what it is and how it is going to comprise of different elements. As you can see, like OpenDevoCell is comprised of four major elements: DevoZoo, DevoWorm AI Center, DevoWorm Academy, and library packages and support. and these are the different uh, types. Like these are the different portions which comprise of these segments. So I'm going to populate it as well as I progress. Next thing is like to-do board. So like in the GitHub repository, the to-do board you get is the project done, which shows like in progress and all this kind of stuff. So like it is very nice, like in the GitHub as well, and we are going to use that. But I want things to be done slightly more good manner. So I want to you people and especially mentors to rate what I'm doing good, what I'm doing so, and what I need to change while doing the project. I don't want to just complete the project. I want to make that project more and more good and engaging. So I will share the link with mentors about this in the over the Slack. So we can put our agendas in the sticky notes here, turn progress and all to do list. So we can also put our weekly agendas here. So this little demonstration like how I'm planning about it. So week-wise timeline execution plan, as I've already stated in my proposal, like week one, I'm going to make different pages. Week one of the coding period, I'm going to make different portals, improving interface of DevoWorm and DevoNotes. I'm starting making static pages. Week two, I've started created dummy dashboard. I'm starting working on improving C.elegance. So like on week two, I started working on improving. Week three is dedicated to C.elegant model improvement, and let's see like if we are able to improve it further. And week four is the work on the C.elegant improvement model and extracting more information from these videos. Like if we are not able to get some good parameters or some good results from the model that I'm trying to build, so we can simply take the finance model, which is obviously very good right now, and start extracting information as much as information as we can. Then we are coming to week five. Week five is the week after first evaluation, I guess. So we start working on the ML model for general biological image segmentation and their improvement through transfer learning. So this is something like, oh, in the biological imaging, you don't always have the images in a particular format. Like there can be very number of shapes, size, and different kind of things. So we are targeting a building. Can we build something general biological image segmentation? That might not be a state of the art for all the models, but it can help the researchers who are trying to just label their images or something like that. So I will start working on that. This is the goal behind it. Feature extraction is completed, like and begin testing of bacillary and other biological models. Start integrating models with the React.js codes. So, and the week 7 start creating the React.js dashboard with a general image segmentation model and two defined models. By the end of this week, this should be completed. Also, we'll start testing models with the DevoWorm dataset. In week 8, in this week, finish up the statistics models for the image segmentation design, connect them to OpenDevoCell as a general statistics tool. So by week 9, I guess our web portal is not completed, but it is ready to be deployed. It depends on like how the other timelines are going, well as good as the other timelines are going. It will be dependent on that pace. So in week 10, I will try to deploy Basileria model like in the open DevoWorm cell. So like segmentation model I already have and I can deploy it easily, but I'm also trying to complete the work which I have left last year because of my academics. So I will try to complete if time allows the Basileria model. If time doesn't allow, so after GSOC I will be working on that. In the week 11, OpenDevoCell model is deployed as Python packages. This is the one of the major goal this year to deploy packages, Python packages. I'm trying to achieve pip as an installer, but let's see. Like probably we will get to it, and it will be a pip install something which we want to name it. Both models are ready to use in the Python codes. Also this week, I will try to upload data set on the Kaggle so that it will be more and more engaging to the people who are listening or who are interested. Last year we also tried to do something like use Kaggle boards and Kaggle terminals to train the model, but Vinay and we hit some rules here. So in the week 12, it is a buffer period. Like I have given buffer period before every evaluation so that I can complete all the remaining stuff, and I have to work on computer cell 3D as well simulations. I am working on it right now in the community period as well, and I will keep working on them as the extended goal this year. So mentor and students sync. So I will be I will be staying in touch with the mentors in this using Slack as frequently as I can. DevoWorm ML meetings. I'm going to give a weekly update about the progress that I have done in the week. Group discussions, like group discussions are the part where the projects are where projects become where projects are converted from good to great. More and more we discuss, more and more we get inputs, and more we are making things right, I guess. So yeah, whenever any one of us hit a hard problem or obstacles, we can just discuss it in a group. So thank you for giving me time to present this. I guess I forgot to introduce myself in the beginning. So yeah, I am Ujjwal. My full name is Ujjwal Singh. I am a second year student at IIIT Delhi. So please let me know if you have any questions or clarification.
Thank you, Ujjwal. Does anyone have any questions? Okay. Well, yeah. So you did bring up something here that I should have brought up before, and that is about the we'll have project boards for each person. So I think Ujjwal has his set up here with Miro, and we usually do it every year in Google Projects. So actually, let me share my screen. Ujjwal, can you? Yeah.
Sure.
So I'll stop presenting. I wanted to show this real quick. So this is what we've done in the past, and this is what we can do by default this year. But let me know if you want to do it differently. So lab meetings, we actually have a list of tasks. So we generate these project boards in GitHub. GitHub makes it pretty simple to set up. And then you would join it, and then every week we'd go through and review things. I'm not sure for Ujjwal if we want to reinvent the wheel on this. So in other words, like if you already have your Miro board, we can work from that. But for Mayukh, I think maybe we need to create a task board if this works.
So like we can always we have to create like a task board in the GitHub. Like it is the best way to keep track of what things are going. Like Miro board is just to give suggestions and improvements. But like there is like some limitations in task board in GitHub. So I guess like drawing workflow and giving suggestion is easier in Miro board. So yeah, that's why I bring it up.
Yeah, so like this is what a project board in GitHub looks like. And so we set it up. We have to do, hold, in progress, and finished. And the advantage of doing it in GitHub is to have it links to different issues. So like I can create an issue here, number 7, number 8, whatever, and I can track it through its progress. We would come every week and we'd review our board, and then we'd like see where we were with different issues. But I can also assign them to different users. So this one I don't have assignments, but I can very easily take like say Mayukh and assign him to a specific issue. And then it tracks all these things. So, it not only tracks it on this board, but it tracks it in the repository. So we would create it for this repository here. And we'd, I'd assign it to each person. And then we'd review it every week. And, with Miro, actually, I like the idea of Miro. The only thing is I don't know if we can integrate it with GitHub directly. I mean, I don't know if we have to, but, well, it's something for Mayukh and Ujjwal to think about if they want to if they want to do things in the project board here, if they want to go with Miro, or if they want to do a combination of the two. Because I don't know if GitHub projects allows you to visualize anything. You were gonna say something?
Yeah, we could keep things flexible for now. We can just like we can use whatever it is necessary at the certain time.
Okay.
Like if it's GitHub that the thing that you showed, if that's what's required, we'll use that. But if we want something that's a little bit more non-linear, because the thing that you showed is a bit linear. So if you want something which has flowcharts and stuff, so then Miro would be better, I guess. Yeah, so we should keep things flexible.
Yeah.
Yeah.
Well, maybe one way to do that is to do like planning in Miro, and then we can actually assign specific tasks on the GitHub.
Yeah, we can formalize it on GitHub after planning it on Miro.
Yeah, that would be good.
Yeah.
So we'll.
Yeah, combination of both.
Yeah,
Ujjwal wrote.
that's good.
Right.
So yeah, we'll do the boards closer to the coding period. So like, be the first week of the coding period, we'll set it up and I'll start assigning things. But actually, it works best.
I've also had a question.
Yeah.
Hello? Yeah, I had a question. Like, can I start pushing stuff before the coding period starts? Like, suppose you send me the data, right? So I've already started working on the data. So I've made some, like, if I start making a pipeline before the coding period, so can I start pushing?
Oh yes, you can.
Yeah, I'll start pushing then.
Yeah.
Because I'll be starting my work tonight. So then I'll start pushing.
Yeah. I mean, it's
Yeah.
like once a week is like the minimum. I just want to make sure.
Yeah, that's fine with me. That's fine with me. I just wanted to ask because I thought maybe it's some formal thing that I have to do it in that specific time. So I just asked to make it clear. Yeah.
Yeah, I think they've encouraged people this year to do this during the community period to like get set up and push some things early. Just in case something happens later in the summer. But I think it's always good to have like.
Yeah, exactly. Because things are like really uncertain right now. Like we don't even know when we'll have our college exams. So like they'll give us a random date and we'll have our exams. So things might not go very according to plan later on. So I want to do as much as possible right now because I have time. I won't have much later on. Yeah, so I just want exactly. Even Ujjwal is saying that his exams are going on right now. We were talking. And so it's kind of hard to balance exams in this, so we want to do everything and we can just document and tell that we have done it during the coding period later on, but we'll just do some of it now.
Yeah.
To adjust, the situation is different today, this year.
Yeah. Well, so I'll set up the board in GSOC 2020 and we'll have that, and then, we'll kind of like when we get to, I don't want to assign really anything yet, but when we get to something that's assignable, we'll start putting up tasks, and then every week we'll kind of review where we are.
Yeah, the mentors can review and assign stuff.
Yeah.
Yeah.
Okay. Well, that's good. So I guess we're probably well over the hour, but I wanted to give everyone a chance to present their work, and we'll have this recorded on YouTube after, so if you want to view it, they can. Otherwise, if you don't have any more questions, Krishna, did you have any questions? Okay. Thank you for attending. Okay. Thank you for attending.
Thank you.
Yeah. And we meet here once a week, every week. So next week, I'm not sure what we're going to have yet. We'll probably have an update from the GSOC students, and then anyone else who wants to update on anything, they're welcome to. So thank you again for attending. Have a good week. Stay safe. Yep, thank you. All right. Bye.
Bye.
Bye.